Hello students, myself Om Prakash Katte and you are welcome to Biology Reward. Today we are discussing the processes of the recombinant DNA technology of class 12 biology, and unit of our topic is biotechnology. So we have to discuss regarding the processes of recombinant DNA technology. Processes of recombinant DNA technology, or it is also called the rDNA, recombinant DNA technology, or rDNA technology. Already regarding the individual steps we have discussed earlier, and now we are summarizing all the steps of the process of recombinant DNA technology. So in recombinant DNA technology, first step is isolation of genetic material, and in eukaryotic genetic material is DNA. So students, we have learned any cell, if you are taking, the DNA is packed inside the cell. So different type of enzymes is used. First one enzyme to dissolve the wall of the cell. If it is wall is made up of it is made up of cellulose. So in plant cell, the wall is made up of cellulose, then their cellulase enzymes are used. In bacteria, wall is made up of polyglycol. In such cases, in bacteria, lysozyme enzymes are used. Similarly, in fungus, these chitinase enzymes are used. So these enzymes are used to remove the wall present in the cell. So cellulase enzymes is used in the plant, lysozyme enzymes is used in the bacteria, and chitinase enzyme is used to remove the cell wall of the cell. After this cell wall, the remaining material left, it is the DNA material plus RNA material plus protein. So in a eukaryotic organism, the DNA is wrapped along with the protein. So here, for the digestion of RNA, RNase enzyme is used. For the digestion of the protein, protease enzyme is used. So due to this enzyme, whatever RNA and protein are remaining in the cell are digested. So only DNA material, DNA material remain left. And if DNA material we are keeping in the chilled alcohol, in the chilled alcohol, the fine fiber of the DNA we are getting on upper surface, and that can be taken out by the spooling process. So this is LR process for the isolation of genetic material DNA. Again, wall is dissolved. If wall made up of cellulose and lysozyme, chitinase, such type of enzymes are used. RNA is digested by RNase enzyme. Protein is digested by protease enzyme. And last one, the DNA molecule can be extracted by keeping them in the chilled alcohol. So fine fiber of the DNA remain left, and that spooling process we are taking out. Second process of the recombinant DNA technology is amplification of the DNA or gene of interest by using PCR method. In such cases where the DNA is isolated, this DNA molecule is isolated. So maybe amount of the DNA molecule required for our different purpose required large quantity. In such cases, either the gene of interest we used to say, that is means desired piece of the DNA. So this desired piece of the DNA is in less quantity. So, but we require more quantity of the desired piece of DNA, and that is called genes of interest. It means DNA amount is very little amount, and if we require in large scale, then this DNA by the method of PCR, polymerase chain reaction method, we are making the millions of copies of the DNA material. So making the DNA copies by using the polymerase chain reaction, this is called amplification of DNA or gene of interest by using PCR, and it is the second method of the recombinant DNA technology. After this method, whatever DNA we are extracting, this is the molecule of DNA, and this molecule of DNA we have extracted. Similarly, cutting of DNA, vector DNA molecule, just the DNA molecule. So one is the molecule of desired piece, as I am indicating it. This is the DNA molecule. This is one DNA molecule I am extracting, and this is DNA, or it's a foreign DNA I am indicating as a foreign DNA. So foreign DNA, it means DNA of interest from which we will get the product, and same we have to extract from the vector DNA. This is the vector DNA. Our vector DNA are foreign DNA. So cutting of DNA of vector and vector DNA, cutting of DNA and vector DNA. So this type of foreign DNA and vector DNA is isolated. Now this DNA, foreign DNA, is cut due to the action of restriction endonuclease enzyme. This vector DNA is also cut by the restriction endonuclease enzyme. So when the foreign DNA is cut restriction endonuclease, it cut the DNA into the some specific sequence, and which is we call it is palindromic sequence. Similarly, RNA, recombinant DNA, REN, recombinant restriction endonuclease enzyme. It is restriction endonuclease enzyme. It cut the vector DNA also in specific sequence. And after that, we are getting a piece of foreign DNA or desired DNA. This is the piece of desired DNA. And it is a piece of vector DNA. So this DNA are cut with the DNA molecules are cut with the help of restriction endonuclease enzyme, and this process where DNA molecules are cut, it is called the cutting of DNA and vector DNA for recombination. Third process after this cutting, these two molecules, foreign DNA piece and vector DNA piece. So formation of recombinant DNA using the ligase. So what happened now? These are both DNA molecules. Fragments of DNA are connected with each other. So this one, when this piece of vector DNA and this foreign DNA, they are connected with each other. So this process is known as ligation process. In this process, it is proceed for ligase enzyme. Here ligase enzymes is used. So with the help of the ligase enzyme, this foreign DNA or gene of interest and vector DNA, they bind together, and this DNA we are getting, it is called recombinant DNA. So this is the process where formation of recombinant DNA takes place. Fragments are isolated. Isolation of DNA fragment by gel electrophoresis. Let's first you should understand. This DNA, foreign DNA is cut into restriction in the presence of restriction endonuclease enzyme, and due to this one we are getting the small fragment of the DNA. Before this process I am explaining one more thing. So when the fragments are cut, these fragments we have to isolate, and for the isolation we are using the gel electrophoresis process. Here also gel electrophoresis. In this process, this small fragment are mixed with the agarose gel, and when they are mixed with the agarose gel, so different strand of DNA are extracted here. This different strand of the DNA or fragments are extracted here. Now these fragments are the foreign DNA fragments, and this is the fragments of the vector DNA. So these fragments, in the presence of ligase enzyme, they are combined together. So this will be the new formation of recombinant DNA, and this DNA is called the rDNA molecule. So after isolation of DNA, DNA is simplified. Then DNA is cut into the DNA as well as vector DNA is cut into specific site, and isolation of fragments by the gel electrophoresis is done. Then this piece of the foreign DNA and vector DNA are combined together in the presence of ligase enzyme, and we are getting the recombinant DNA. Now this recombinant DNA is introduced into the host cell. It is the host cell where we have to introduce this recombinant DNA. So for this purpose, insertion of recombinant DNA into competent host organism. So competent host organism, we have learned how to make the host competent. So treating with the divalent sign or gene gun method is used. Sometimes we are the non-pathogenic or non-distant pathogen is introduced. Through this one, this recombinant DNA is introduced into the host cell, and that is called insertion of recombinant DNA into the organism. So once the recombinant DNA is inserted into the host cell, now the host cell is ready to give the product of the foreign gene. So by this host we are obtaining the foreign gene product. So we will get the recombinant proteins, we will get the different type of antibiotics is for the purpose is done. So we are obtaining the product of this foreign DNA, and this foreign DNA, if we require product in the large scale, so large scale production of this product or recombinant proteins is made in the bioreactor. So bioreactor, what happens in this bioreactor are used for the commercial or large scale production of the recombinant protein or foreign genes product. In such cases, some vessels are used. In this case, the large scale production is done. So it is called the bioreactor. After bioreactor, we are getting the large scale production of recombinant DNA product. So bioreactor are used for the production of large scale production of the product of the recombinant DNA or this product of the gene of interest. After the large scale production, then this product we have to send for the marketing. And for the marketing, downstreaming process is used. So what is downstreaming processing for the marketing? So before sending into the market, first one, whatever products we are getting, these are the product, large cell products are formed. In this large cell product, the first process is done, it is separation. Undesired material are separated from the desired product. So when production is done in the large scale, so many unwanted product also are formed. So these unwanted product are isolated or separated from the desired product. Then purification. If there are any impurities, that impurities are also taken out. And then testing for the quality control. Whatever product, suppose the alcohol-like products are obtained. In such cases, the quality of the alcohol is tested. So the quality control testing, and last one, if it is confirmed that the product obtained are desired quality, then they are packaged, they are packing, and these are sent to the market. So in again recombinant DNA technology, thus we are having isolation of DNA. That DNA which is isolated, it is genes of interest, and it is amplified, large number of copies are made. So DNA which we are obtaining, we have to cut This is the cutting of DNA and vector DNA. This whatever DNA we are cutting, the DNA fragments are separated by gel electrophoresis, and then cutting of DNA vector and vector DNA, it means their fragments are combined with the ligase enzyme, and it is called formation of recombinant DNA technology. Then insertion of recombinant DNA into competitive host. This recombinant DNA form, then it is inserted into the competitive host. Then we are getting the foreign gene of product. Then these products are done in large scale with the help of bioreactor, and then it is proceeding for the marketing. So it is called downstream process, where separation, purification, quality testing and packaging is done. So it's generally in our book the isolation of DNA and amplification of DNA, then cutting of DNA and vector DNA. This after cutting, formation of recombinant DNA, then insertion of this one, foreign genes product, large scale production and downstreaming. This process are explained together in other parts. Here it is explained isolation of DNA fragment by the gene electrophoresis. So if recombinant DNA technology question is asked, then we have to describe in such way. It's all about the recombinant DNA and its process. Thank you.